Membrane proteins don't exist in a vacuum, but when you open one in PyMOL, that's essentially what you see, a protein floating in three-dimensional space. You don't immediately know where the membrane should be, which residue sits in its core, whether something unusual deserves inspection, or whether two different membrane orientation sources actually agree. So I built a PyMOL plugin to make that review process easier. It's called Membrane Visual QC, and version 1.0 is finally finished. What started as a relatively small visualization script eventually became a much larger project about membrane geometry, provenance, scientific interpretation, failure handling, testing, and reproducible software releases. So in this video, I don't just want to show you how the plugin works. I want to explain why I built it, what scientific idea sits underneath it, and what it looked to turn into something I was actually comfortable calling version 1.0. Let's start with bacteria rhodopsin. PDB is 1M0L. It's a compact 7-transmembrane protein from Halobacterium salinarum, built around a retinol chromophore and functioning as a light-driven proton pump. The structure we're looking at was determined at 1.47 angstrom resolution. And visually, it's a great membrane protein example. You can clearly see the helical bundle, but there is still a problem. Where is the membrane? The coordinates themselves give us the protein. They don't automatically give us the biological context in which we want to inspect it. The simplest workflow uses an explicit membrane frame. For this demonstration, I'm using the global Z mode, with boundaries at minus and plus 15 angstrom. Those values are demo parameters. I'm not claiming that every membrane has exactly this thickness. I select the protein and run the QC. The important thing is that I'm not asking for one magic score that says the structure is good or bad. I'm asking geometric questions. Where are the residues relative to the membrane? What deserves attention? And can I make the context immediately visible? When I show the slab, those abstract coordinates suddenly become much easier to interpret. Now I can actually see the protein crossing the membrane region I'm using for the analysis. Geometry alone isn't enough. Membrane proteins are interesting partially because residue chemistry and membrane position are connected. So the plugin can color the structure by hydropathy. The idea of assigning amino acids hydropathic character is not something I invented for this project. A classic reference is the Kite-Doolittle hydropathy scale, published in 1982 and still widely used as a way of describing hydrophobic and hydrophilic tendencies in protein sequences. What I'm doing here is much more modest than membrane prediction. I'm using chemical information as a visual inspection layer on top of already known three-dimensional structure. Now, unusual chemistry in a membrane-facing region is much easier to notice. But notice the wording. Notice, not automatically condemn. A polar or charged residue in a membrane region may be biologically important. The software should surface for its inspection, not declare it wrong. Bacteria rhodopsin also gives us a useful local example, retinol. Instead of asking about the whole protein, I can ask a much more focused structural question. What is close to this ligand? Here I select retinol specifically and build its local neighborhood at a defined distance cutoff. Again, the cutoff is a parameter of the analysis. It isn't evidence by itself that every residue inside the distance is functionally important. What the plugin gives me is a reproducible, structural neighborhood that I can immediately inspect in 3 dimensions. I also didn't want Membrane Visual QC to be just a collection of buttons that produce a nice picture. Every analysis can be exported as a structured JSON. That means the visual review can leave behind a machine-readable record of the parameters, orientation, result, and provenance used in the analysis. These became especially important as the project grew. If I inspect a structure today, I want to be able to determine later what version of the reporting contract produced this result, which source defined the orientation, and what the program actually observed. Version 1.0 also uses the strict JSON output. Non-finite values like none or infinity cannot silently leak into a report that is supposed to be reproducible and machine-readable. And membrane proteins obviously aren't all 7 helix bundles. This is OMPF, the outer membrane pouring from E. coli. Each OMPF monomer forms a 16-stranded beta barrel surrounding an aqueous pore, and the protein normally assembles as a trimer. Visually, this architecture is completely different from Bacteria-Dopsin. And that's exactly why I wanted the central geometry of the plugin to be defined relative to the membrane frame rather than around one specific protein architecture. The same basic question still makes sense. Where is the structure relative to the membrane? This leads to one of the parts of the project I became increasingly careful about: provenance. A membrane orientation isn't simply the membrane. It came from somewhere. PDBTM, for example, is a database built around identifying transmembrane proteins from known structures and locating their membrane-spanning regions using 3-dimensional structural information. Membrane Visual QC can consume validated PDBTM-derived data, but it doesn't simply trust 2 files because their names look related. It checks their structural relationship. Here, the mapping resolves through the inverse provider transform. 12 atoms are matched with 0 NMSD and 0 maximum residual in this retained validation example. So now the membrane orientation isn't just a visual assumption. We also have evidence about how the provider-derived structure maps back to the structure where we're analyzing. At this point, I realized the project was becoming something slightly different from what I originally imagined. It wasn't just about making one structure easier to inspect. If the workflow was going to be useful beyond a single example, I needed to think about repeatability, multiple structures, failure states, and provenance at scale. That's where batch review came from. A batch plan defines the analysis before they run. Here I have 5 jobs covering several orientation workflows and a source comparison case. The plan is validated first. Then, the jobs execute using the same explicit configuration rather than manually repeating a sequence of clicks and trying to remember what I did. And the outcome vocabulary is deliberate. A job may complete successfully, and input may be rejected. Or the analysis may produce review items. A review item does not mean this protein failed. It means the analysis completed, but something is worth a human look. This distinguish become one of the most important design boundaries in the whole project. Another batch compares orientations from PDBTM and OPM. OPM is a separate resource that provides special arrangements of membrane proteins relative to the lipid bilayer. And this is where wording matters a lot. Membrane Visual QC does not ask which database is better. It asks, can these 2 orientations be compared geometrically, and if so, how different are they? In this retained example, the sources are comparable, but the result fails into a measurable geometric difference band. This is not a ranking. It is not evidence that one orientation is biologically correct and the other is wrong. It is a reason to inspect the difference. The funny thing is that by this point, the scientific logic was no longer the hardest part. The difficult question became, what would it actually take for me to trust this as a software? Not just on my machine, not just one structure, and not just when everything goes right. That meant testing things I absolutely wasn't thinking about when I wrote the first version. Malformed input, Windows reserved file names, Unicode paths, paths containing spaces, batch cancellation, recovery, strict serialization, clean installation, upgrade and rollback behavior, and normal using mistakes that should produce useful messages instead of Python tracebacks. By the final release, the project had more than 1,000 automated tests, with CI across multiple Python versions, Windows-specific checks, and independent reference validation. I also didn't want version 1.0 to mean I changed the version number because the interface looked finished. Version 0.9 became a real pre-release and went through a clean, install, and upgrade testing. Then I published 1.0.0 release candidate 1. That candidate went through another real PyMOL acceptance run. After the remaining hardening work, I repeated the acceptance against the actual stable 1.0 artifact, including upgrade from the release candidate, compatibility checks, rollback, and final reinstall. Only then I did publish version 1.0. The stable release is now available directly from GitHub. The plugin zip is built deterministically and published with a SHA-256 checksum. After publication, I downloaded the release asset again independently and verified that it was byte identical to the artifact that had passed the final acceptance. The release also used an annotated git tag and frozen release evidence. That still isn't the same thing as code signing, SIG-store attestation, or a cryptographically authenticated build pipeline. And I don't claim that it is. A checksum verifies integrity against a trusted digest. It does not by itself prove publisher identity. For me, that distinction is exactly the kind of thing scientific software documentation should be explicit about. And that brings me back to the most important design decision in the project. Membrane Visual QC is not trying to replace scientific judgment. It doesn't look at the structure and declare, this protein is correct. Its job is more narrow. Put the structure into membrane-aware geometric context, make interesting chemistry easier to inspect, keep track of where the orientation came from, surface review items, compare orientation sources when that comparison is meaningful, and leave behind enough structured information that the analysis can be reproduced later. A scientific tool should make evidence easier to reason about. It shouldn't pretend uncertainty doesn't exist. So that's Membrane Visual QC 1.0. What started as a relatively simple PyMOL script turned into a much bigger exercise in structural bioinformatics, scientific provenance, software design, and reproducibility. The project is open on GitHub and version 1.0 is available now. If you work with membrane proteins, structural bioinformatics, protein design, or you simply use PyMOL a lot, I would genuinely love to hear what you think. And if you try it on structures beyond the examples I've shown here, I'm especially interested in what works, what doesn't, and what would actually be useful in a future version. The repository and references are in the description. Thank you for watching.